Right, okay, we're going to walk through some polymers, amino acids, and DNA questions here. To start with, we've got this polymers question. We've got two repeating units here, P and Q. Firstly, let's do this one. We've got to draw the structure of the monomer used to form polymer P. So the way I would approach this is to go back from the repeating unit into the monomer. If we effectively just cancel both of the dangling bonds on either side and then reform the double bond in the middle like that, we'd have our monomer. So it would be this carbon, carbon, hydrogen, CH3 down there, Cl, and then another CH3. Okay, right, I'm going to skip ahead a little bit and look at this question. Very common question this in polymers. 2 or 3 marks usually suggest an environmental advantage of polymer Q over polymer P. So Q over P. So big thing here is looking at the type of bonds that we've got in there. All right. Polymer Q in particular, we've got this polar C double bond O, which means that it's susceptible to attack by nucleophiles. So that's the what I would say in this answer. The advantage there, the idea is that it's biodegradable, right? So polymer Q is biodegradable, and the justification, the polar carbonyl bond, C double bond O bond, is susceptible to attack by nucleophiles. Okay, even something like water, quite a low rate of reaction nonetheless, but still can be a nucleophile. Okay, so we've got this question here about amino acids. So we've got a diagram showing two parts of 2 polypeptide chains in a beta-plated sheet of a protein. So alpha helices and beta-plated sheets are joined through hydrogen bonds, which we can see there and there. The polypeptide chains are held together by hydrogen bonding, as shown in the diagram. So these are the dotted lines that you see there and there, for example. And it says, explain how these hydrogen bonds form. And the marking is really kind of tight on this, okay? So you've got to use the exact right terminology. What we want to say here is that if we look at this hydrogen bond here, for example, what you want to say is that there's an attraction between the lone pair on the oxygen and an electropositive hydrogen, right? They really want to hear that language, okay? So, say there is an attraction between the lone pair on O and a delta positive hydrogen atom. Okay, so that's the language that they want to hear for those 2 marks. Okay, a different type of bond can form between 2 polypeptide chains when the chains each contain the amino acid cysteine. Complete the structures to show the bond that forms when the side chains of 2 cysteine molecules. Okay, so here we go. So they've drawn parts of the structure of 2 cysteines, and they want us to finish it off. Okay, so if I bring in a data sheet to help us a little bit here, okay, and I'm going to be looking at cysteine in particular, which we've got up here. Now, it's the side chain that is of interest, so it's the CH2SH bit. Okay, so what we want here is, we've got this CH here, but look, this is the CH between the carbonyl and the amino groups. So that's this CH here. Okay, so what we want to do is we want to have a CH2 first of all, and then we want to have the S, right? So we want a CH2 first, there, and here, let's say. And then after that, we're going to form our disulfide bond. Okay, there. All right, the 2 hydrogens get eliminated. We don't need to draw them. Okay, when we form the disulfide bridge, those 2 hydrogens get eliminated. Okay, so next one here, draw the Zwitter ion of a dipeptide formed by alanine and serine. So alanine and serine is what we're after here. So I've got them both in view here. There's alanine up there, and I've got serine down here. Okay, quite difficult to get everything into shot, but nonetheless. Okay, so what you want to do is there's two possible answers here, depending on which way around you connect them. Okay, so I'll start off with the alanine. I'll start off with the amino group, but I want to keep in mind that I want to make the Zwitter. I want to draw the Zwitter ion. All right. So we're going to start with alanine. I'll start with the amino group. So the amino group is NH2, but in the Zwitter ion, the amino group is going to be protonated. So I've got to protonate the amino group on one side. So there we go, and put a positive charge on the end there. Okay, and then I'm following through now with the rest of the alanine molecule. So CH3, and then here I've got the carboxylic acid group. But I'm obviously not going to draw it completely. I'm going to have the carbonyl C double bond O, but I'm going to lose the OH because this is where it's formed the bond, the amide bond to the serine, or peptide bond if you prefer. Okay, so C double bond O there from the alanine, and now we're on to the amino group of the serine. So find the serine, there it is. So there's the amino group, and then the rest of it now, CH, CH2OH, no deprotonation down here because it's an alcohol group, doesn't really undergo deprotonation. And then here's where we can have deprotonation, is at the very end where the COOH would be. It's actually now COO minus, like that, because the proton has been lost there, but gained there if you like. Okay, so there we go, that's the Zwitter ion. Skim ahead, see what else we can pick out from this. So this question here about amines, quite a nice little question about amines down here. So we want to complete the mechanism for the reaction of ammonia with 6-bromohexylamine to form 1,6-diaminohexane. Okay, so they've started the mechanism off for us. We've just got to complete it. So it's a lone pair on the ammonia, attacks this carbon here. They've made it skeletal, that's the complexity that they've added, but this is the carbon that we're attacking, so it's that arrow first of all. Okay, now from then on in you've got to complete it, so you don't have to follow it through skeletally. You can draw it out in full now if you like. It's a bit more up to you. So I'll have the NH3 on. Okay, positively charged nitrogen there now, and then it's bonding to this carbon, so 1, 2, 3, 4, 5, 6 carbons. So I'm going to shorten this slightly and just say CH2 6, just to use a bit of shorthand to help me out, and then NH2. Okay, and finally then to finish it off to form 1,6-diaminohexane, what do we need? So we need an excess of ammonia, and then the lone pair on this ammonia will take this proton, and the electrons from the NH bond here will fold into that end there. Okay, and if you wanted to, you could draw out the final structure, which would be something like this. Okay, then another part of this question suggests the structure of a cyclic secondary amine that can be formed as a byproduct in this reaction. So in the original mechanism, which they gave to us to do, we've got the carbon being attacked by this NH3, which is effectively it's an attacking species. It's an attacking nucleophile, right? But what could potentially happen is this. Let's draw this out again. So I've got my Br, then I've got my chain. There we go. Now, remember what's happening here. The bromine is partially negative and this carbon here is partially positive. So it's prone to attack by a nucleophile. Now, there is another nucleophile present in this molecule. This nitrogen, this amine, nitrogen atom's got a lone pair, which will be attracted to this partially positive carbon center. So imagine instead of this nucleophile attacking, what about if this nucleophile attacks? If we follow this mechanism through in the same way that we did up here, okay, what you'd end up with is a cyclic secondary amine, which would look a bit like this. Okay, so all you've got to do there is just make sure that you add the H there and the correct number of carbons. So there's you're forming a bond between this N and this carbon here. So 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6 carbons. So this would be a correct answer for the cyclic secondary amine. Okay, very common question they like to ask about amines is this type of question. Explain why 3-aminopentane is a stronger base than ammonia. So if I go downstairs really quickly, here's your ammonia. Forgive the inaccuracy of the drawing. Just want to get the idea that we've got a lone pair on that ammonia, okay, which makes it basic. Okay, think of a base as a lone pair donor or a proton acceptor, and it's to do with the availability of this lone pair to be donated or to accept a proton. So let's compare this then with this 3-aminopentane. So pentane is 5 carbons and it's 3 amino, so got the amine group in the middle there. Okay, alright, so we've got here CH3 group, CH3 group there. This is a CH2, this is a CH2, and this is just a CH. Alright, so the key thing here is that let's put the lone pair on the nitrogen there. What we've got here is we've got an alkyl group attached. Okay, so what we've got is an electron donating effect, a positive inductive effect. Electrons are being pushed towards the nitrogen atom, meaning that this lone pair is more available to donate. Okay, so that's our answer really. Okay, so in 3-aminopentane, the alkyl group attached to N, you could say that it provides a positive inductive effect. Okay, and this makes the lone pair, so I'm going to say making the lone pair on nitrogen more available to donate. And like I said, it's a very common amines question in organic A-level chemistry. Okay, there we go.